Good evening, all. On behalf of the organizing committee, I, Srindanti Ghosh, welcome our Principal Madam, IQAC Coordinator Madam, honorable speakers, participants from different colleges, and our lovely students in the second session of the one-day international webinar on recent trends in global health and healthcare, organized by Departments of Life Science of Hooghly Women's College, West Bengal. Now I am requesting Dr. Shamolina Goswami, the IQAC coordinator of Hooghly Women's College and the advisor of the webinar, to say few words.
Thank you. Thank you, Srindanti. Uh, we have come to the second part of the today's international seminar. I welcome on behalf of my college and the seminar with topic recent trends in global health and healthcare. It's the topic of today's. It gives me a pleasure and tremendous contentment to presenting the welcome speech among the most esteemed personalities, the scientists from different parts of the world. Thank you so much. On behalf of my college, I would like to request all the participants to stay throughout the session. Thank you so much. And once again, I would like to welcome all for finding time and visiting today's international seminar, webinar. Thank you so much. And
Thank you, madam, for your beautiful speech, and please stay with us. Now I am requesting Dr. Shima Banerjee, our respected principal of Hooghly Women's College and the patron of the webinar, to say few words.
Good evening, friends. Today we are being assembled here to attend an international webinar organized by all the departments, departments of Hooghly Women's College. We have two parts, two sessions of this seminar today. In the morning session, we have enriched ourselves about COVID-19, the new threat to the mankind. Nearly 213 countries of the globe are being affected due to this unforeseen enemy of human beings. Now, in the second session of today's international webinar, we have two eminent speakers who will enlighten us about two well-known and frequently shown diseases of human life, heart-related problems, and cancer. As in modern times, nearly most of us have to lead stressful life due to our profession, and that is why our lifestyle are also affecting. So cardiac problems are very common feature. Most of us are suffering from hypertension. Similarly, people are suffering from different types of cancers too. The number of cancer patients is also increasing rapidly. Maybe the changing pattern of lifestyle accompanied with different types of changing nature of jobs, continuously changing dietary habits, and even ever rising events of exposure to pollutants are the main causes. Two eminent speakers dealing with these two above mentioned two fields will provide us now new ideas to prevent the diseases, and hope is that definitely we will be familiar to the continuously going efforts to discover remedies for these two major health hazards. Thanks you all once again, and over to Srinunti.
Thank you, madam, for the informative speech, and please stay with us. Now it is over to Mr. Ashok Murmu.
Thank you, ma'am. Good evening to all. In this international webinar, recent trend in global health and healthcare. In second session, we are blessed with two eminent speakers. One is Dr. Manoj Kumar Pal from University of California, USA, and his discussing topic is stem cell research to cancer drug discovery. And second half will be enlightened with Dr. Nandan Kumar Mandal, Assistant Professor, Baylor College of Medicine, Houston, Texas, USA, and discussing topic is current scenario and failure, future perspective of heart failure, biomedical devices, and heart transplantation. Now again, I would request Mrs. Srinanthi Madam, kindly introduce our first speaker of first session. Thank you.
Thank you, Ashok Babu. As we all know that our first resource person is Dr. Manoj Kumar Pal from the University of California, Los Angeles. Before starting the webinar, I want to introduce him before you. Dr. Manoj Kumar Pal is an expert in the field of stem cell and cancer and currently working as a scientist at the University of California, Los Angeles. He has contributed significantly to the field of stem cell biology, regenerative medicine, and lung cancer. He has revealed several drug discovery targets involved in maintaining stem cell homeostasis during injury and repair and also contributed to drug discovery. He is an expert in organoid biology and disease modeling of the lung. He, along with his group, has also published first in vitro method for the development of lung organoids mimicking lung from iPSC. Dr. Pal is the recipient of many awards that include the prestigious UCLA Vice Chancellor Award and was nominated for the Pal Boers Award. Dr. Pal is a member of many scientific societies like ISSCR, AHA, ALA, and editorial member of many international journals. Dr. Pal is passionate about using new technologies for stem cell-based therapy and regenerative biology. In this COVID-19 pandemic situation, a team of scientists has developed a machine for sterilizing PPE kit to all types of medical equipment and other domestic and personal materials. The team is led by Dr. Manoj Kumar Pal from the University of California, USA, and Professor Dr. Rajiv Banerjee from Dr. B.C. Roy Engineering College, West Bengal, India. So far, he has 39 publications, including research papers, reviews, and book chapters, etc. We are happy to offer him heartfelt warm gratitude and we are thankful to him. Now, it is over to Ashok Murmu.
On behalf of organizing committee, I want to request Dr. Pal. Proceed now. Dr. Pal, you can proceed now.
Hello, can you hear me?
Yes, I'm hearing you. Yes, you are audible. Audible, sir.
Okay, I will project my screen. Give me a second.
So, is there any problem, sir?
I'm trying to project my screen. This I'm not getting it yet. Yes. So is my screen visible?
Yes, yes, sir.
And can you hear me?
Yes, of course.
Good.
Clearly.
So good evening, everybody. It's
Carry on.
a real pleasure to give a talk at this prestigious webinar series. And I really appreciate Dr. Shamalini, Dr. Seema, Dr. Simanti, and Dr. Ashok for this nice introduction and giving me this opportunity to present my work with you. I work in the area of stem cell research, regenerative medicine, and cancer. And I'm presenting today some data looking at the role of wind signaling dynamics in the airway in the context of regeneration and cancer. So the lungs are complex and vital organs and can be divided into the proximal airways and the distal lung. So the proximal airways are these, and the distal lungs are on this side where you see the alveoli, which are mostly involved in the gaseous exchange, while the proximal airway, as you can see here, which you call trachea, is lined by epithelial. This is a cross section of that, and you can see these are lined by epithelial layer. And this epithelium looks like this. And these epithelial cells produce mucus as well as their tiny hair-like cilia, which which undulates and produce a synchronous beating, as you can see in the video, to move the mucus layer in which harmful particles and debris get entangled. And so this is the way the mucociliary clearance happens. And this is very important for the immunity, because if the mucociliary clearance is blocked, then the bacteria and virus and other particles can sit on the epithelium and then infect the epithelium, and then infection starts. So how do we study injury in the trachea? I work mainly on the trachea, and how do we study this injury in the tracheobronchial epithelium? So the tracheal epithelium is very susceptible to injury because the whatever we inhale goes directly and falls on the epithelium and can cause injury. It can be cigarette smoke, it can be harmful gases, it can be anything, and it causes injury. So how this we can model in mice, how to model this injury in mice is a question. So we used a method called polydocanol based injury model, where we use polydocanol. Polydocanol is a surfactant. When we use intratracheal injection of polydocanol, then polydocanol sloughs off the epithelium. The epithelium looks wild type uninjured epithelium looks like this, and where K5 marks the in red the stem cells, and some ciliated cells, as you saw in the video, these ciliated cells are marked by acetylated beta tubulin, and DAPI is marking the nucleus of all cells. And if we create a polydocanol based injury, then what we find is that the K5 cells are only left out, or other cells are gone. So from this, the stem cell starts proliferating, and the stem cell start proliferating at first by using symmetric division, and after some time, and leads to the symmetric division leads to more K5 positive cells, and after some time, it starts doing asymmetric division. In asymmetric division is going to the apical basal way. The angle of the poles of the spindles are like this, and then it creates the early progenitor cells like K8 marked by cytokeratin 8. And you can see slowly with time, it's a time course of injury repair as shown is here. The proliferation becomes maximum at 48 hours, and after that the differentiation phase sets in, and around 96 hours the first differentiated cells starts coming. So differentiated cells are marked here by CC10, which marks the club cells. I'll come back to that again. And you can appreciate that at 120 hour around that time the differentiation has initiated and is going on, and ultimately it differentiates into a proper looks like uninjured trachea. uninjured tracheal epithelium. But what happens if this injury and normal repair is becomes aberrant? And in that case, the stem cell proliferates, and they create a stratified epithelium, which looks like a pre-malignant epithelium. And then once the hyperproliferation increases, and these cells, the stem cells, the K5 cells, then invaginates the basement membrane, then it like is considered as the hallmark of malignancy. So in our lab, we try to figure out how this normal repair process happens and how that normal process is affected in pre-malignancy and other diseases. So I will bring like show you the proximal airway lineage hierarchies, which is what type of cells are there, how the stem cell hierarchy works here. So again, on the left side, I'm showing you the club cells, which are important cells. As you can see here, the stem cells, which are shown here in green on the left-hand side, and the stem cell is shown here in blue. The stem cell can differentiate into club cells. Here, the club cells are shown in red, and the stem cell can divide in form ciliated cells or tuft cells, and these are shown here in yellow. So these stem cell itself can repair the whole airway epithelium, but it takes some time. And aberrant like defects in that proliferation can cause cancer. So the point is, what is important here is the basal cell, the club cells, and the ciliated cell. These are the most important cells which are important. Other than that, there are different other differentiated cells and which play a role, but these are the most significant ones. So we will be studying these in details. So the airway basal stem cell, and what is the putative niche? And the question is, what is a niche? Niche is a microenvironment which helps. Niche is a microenvironment which helps the stem cell, the airway basal stem cell, we call them, to like maintain the stemness as well as differentiate over time, so that the stem cell over the whole adulthood remains there and can respond to injury and repair the whole epithelium. And there can be other niches. The stem, what are the niches? So niches can be this intracartilaginous zone, because in the trachea, you all know there are cartilages. These cartilages help to protect the trachea from collapsing. When we breathe, it can collapse, but the tracheal rings around the trachea, the cartilage rings protect it from collapsing. And in between the cartilages, there is a intercartilaginous zone which is made up of different type of stromal cell, which includes fibroblasts, endothelial cells, and immune cells. They also, and neurons, they also play a significant role and can act as a niche for these stem cells. So to understand what is the role of wind signaling in airway, we started working on wind signaling because wind signaling is not much worked out in airway epithelium, and moreover, wind signaling is aberrant in certain squamous cancer. So to understand what is wind signaling and how it operates in the airway epithelium, we started working on wind beta catenin pathway. So I'll just brief you on how the wind beta catenin pathway works. There is a complex of proteins which phosphorylates beta catenin, and once it's phosphorylated, the beta catenin is ubiquitinated by beta TRCP. The beta TRCP ubiquitinated is, and once it's ubiquitinated, it's meant for degradation through degradation, and the beta catenin is lost. So in absence of active Wnt ligand, there is a degradation of the beta catenin and loss of beta catenin, and Wnt responsive genes are inactive. So what happens when the Wnt signaling is on? So the Wnt is secreted. The Wnt is secreted, and then the Wnt needs a protein called porcupine, which uh, which induces post-translational modification of the Wnt, and post-translational modification of Wnt, and the Wnt is then secreted outside the cell. Once it's secreted outside the cell, it can bind with a ligand called Frizzled and the co-receptor LRP56, and then this destruction complex, which was earlier destroying the beta catenin, cannot work anymore, and the beta catenin becomes active and free in the cytoplasm, and then it can go into the nucleus and undergoes transcription of Wnt responsive genes. So now the Wnt responsive pathway is active once the Wnt is secreted from the cell. Now this Wnt can come from the same cell and bind to the same cell as an autocrine signal, or can go to some other cell and bind somewhere else in another cell and can do a paracrine signaling. Now, we wanted to know what is the role of Wnt beta catenin signaling in airway repair. So what we did is we used a polydecanol mediated intratracheal injury, like I explained the injury model, and then we sat the mice at different time points. And before that, we injected these mice with BRDU to study the number of cells which has the BRDU incorporation. The BRDU is bromodeoxyuridine, and it incorporates into the dividing nucleus, and we can detect it. So therefore, we can see how many cells are dividing at a particular time using BRDU. And you can see from this that at 48 hours after injury, uh, the proliferation rate is maximum, and it's before and after that it becomes slow, and it becomes slow. So to understand what is the role of beta catenin signaling during airway repair, we induced the injury and we allowed the repair to happen over time. At the same time, we stained for phosphobeta catenin phosphorylation forms. And we found out of the reported beta catenin phosphorylation forms, none of them is very good in airway epithelium. And we found a new phosphoform, which is called beta catenin Y489, and this Y489 goes to the nucleus, as you can see, with the injury repair. While the proliferation is maximum at 48 hours, the nuclear localization of beta catenin is maximum. And as I said earlier, beta catenin, once it goes into the nucleus, it can activate the Wnt responsive genes. And the repair, with the repair, the slowly as the injury repair is happening, the Wnt beta catenin goes away. The beta catenin Y489 goes away. That means this Y489 is very much associated with the proliferation phase. So we wanted to know whether Wnt signaling play a role in the repairing airway in vivo. In vivo means within the mice. So we planned an experiment where we did an experiment with K5-3ER. So we wanted to remove the beta catenin from the stem cells only and no other cells in the mice. So what we did, we used a K5-3ER tamoxifen inducible reporter line along with beta catenin floxed mice. So this cross, what will happen to these mice is, this mice, the beta catenin will be deleted from the stem cells from here. And sorry, let me put on this laser pointer so it will be easy. So then we wanted to know what is the role of beta catenin. If we delete the beta catenin within the stem cell itself, what will happen? So beta catenin is deleted because we have used a floxed line and with K5-3, and what we see is, this is the experimental plan. When we used tamoxifen before the experiment was done and before the injury was done, and then BRDU was used as earlier to study the proliferation rate. And as we can see in the uninjured epithelium, there is no difference in the epithelium. So that means these mice were not affected before. And after the injury, we study at 48 hours, and we can see significant amount of BRDU incorporation in the normal mice, and but not that much in the transgenic mice, because this epithelium is attenuated. This stem cells cannot divide. This suggests that the Wnt ligand secretion is important, and Wnt is important for proliferation. Now, the question is important is, Wnt ligand secretion is necessary or not? So we studied this using this inhibitor called LJK974, which is the inhibitor of the protein porcupine. As I said earlier, that porcupine adds a lipid tail to the Wnt, and once it adds this, Wnt can go out of the cell and then can bind with the ligand and make the Wnt pathway active. So this LJK974, this inhibitor inhibits the porcupine, and therefore Wnt cannot go out and cannot activate the Wnt pathway. We wanted to know what is the effect, or is the effect the same as that we found with beta catenin? And we found with this porcine inhibitor that yes, after 48 hours of injury, we saw significant amount of proliferation in vehicle control. And because we are using a small molecule inhibitor, we use PBS, phosphate buffer saline, as a vehicle control. And then we saw a significant attenuation of these epithelium in the drug treated ones. That suggests that wind is important for proliferation of these cells. Now, whether the wind is coming from which place, how the wind is coming, we wanted to know. So we stained the porcupine, we stained for porcupine in the airway epithelium during injury repair, and we found that in uninjured mice epithelium, there is no porcupine almost available here or here. But after 48 hours of injury, porcupine increases in the epithelial layer as well as submucosal intracartilaginous zone. And also it slowly wanes out after 72 hours. So that means it again coincides with the 48-hour time point when the maximum proliferation is witnessed. So the question is, now, as we can see, the porcupine is there in these cells, which are probably stem cells, and also in the stromal cells. Now the question is, which compartment is more important and working as a niche for this stem cell proliferation? To better understand that, what we did is we overexpressed, we blocked porcupine, we inhibited porcupine in the K5 positive cells using a K5 porcupine flox transgenic mice. So what is going to happen in this is the porcupine will be deleted from these stem cells only, and not in these cells or not in other cells. Only the stem cells will be deleted. The porcupine will be deleted. This is the beauty of transgenic experiments like this. And so we found that once we stop the porcupine in the stem cells, we don't see a significant difference in the transgenic versus wild type control. And you can see the number of cells dividing here and the number of cells dividing here, and the epithelial pattern of stratification is similar. So it suggests that if we inhibit porcupine, the wind signaling from these stem cells only, it does not create a difference. So the question is, then we quantify the BRDU positive. This also suggests the same thing. There is no difference in significance of the BRD incorporation. Now the question is, then where from the wind signaling is emanating? Is it from the ICG niche? So to study that is this, we plan an experiment where we use the same, we use the global wind secretion blockage by using the LGK 974. Again, we use LGK 974 to study the global porcupine inhibition. And we found when we inhibit globally, means throughout the intercartilaginous zone, the stem cells and all compartments, the stem cells don't divide. That means it clearly suggests that the stem cells don't divide because the wind is not coming from the stem cell but from some other compartment. So what is the identity and the relation of the wind producing niche we wanted to study next? So in intercartilaginous zone is interesting. We found certain fibroblasts which express PDGFR alpha. Now the question is why we are targeting this PDGFR alpha. We searched the literature and found that in intestine also, intestinal stem cell, there is a PDGFR alpha positive fibroblast which can contribute to wind signaling. As intestine and the lungs are endodermal in origin, and they originate in the same way in the during development, we thought maybe there can be some similarity in the signaling aspect also. So what we did is we used a PTGFR Cre Rosa MTTMG. In this mice, what happens is the PTGFR, if it's active, then the cells will be marked by green color. And you can see these are the PTGFR positive cells and the lineage of those cells. And if we used other PTGFR Cre ER and Rosa TMG mice and injured the airway epithelium with polydocanol and studied it after 48 hours. We see with the porcupine is expressed in the airway epithelium as well as the stromal area of the intercartilaginous zone. And then we quantified the number of PDGFR minus porcupine is around 45%, and the PDGFR plus positive and porcupine negative is a small percentage, and PDGFR positive and porcupine positive as another 45%. So it suggests that a significant number of PDGFR positive skeletal fibroblasts can also express porcupine, but there can be another group of fibroblasts or mesenchymal cells which is also producing porcupine, suggesting this can be a niche, and out of that niche, a small fraction, a significant fraction is the PDGFR positive fibroblast. So to understand the lineage necessary for Wnt to drive the stem cell proliferation after injury, so we now, interestingly, we wanted to delete porcupine from these fibroblasts, the PDGFR positive fibroblasts. So other cells will not be affected in the case. Only the PDGFR positive fibroblast cannot secrete Wnt because we are using a PDGFR Cree with a porcupine flux, ROSA TMG. So that means the porcupine will be deleted from inside these cells. And therefore, we can understand the role of Wnt signaling coming from this cell in epithelial repair. And we found that after 48 hours, the uninjured would look similar. So there was no difference in the uninjured epithelium. But after 48 hours of injury, what we found that the BRD incorporation is similar. And but in this case, if the porcupine is blocked in these PDGFR positive cells, we don't see a significant stem cell proliferation. So, and the data is here, and the BRD incorporation is also significantly low, suggesting that clearly that the ICG intercartilaginous zone PDGFR positive lineage is a critical source of Wnt ligand, and that is important for stem cell proliferation following injury. So, if we summarize this, we can say that our experiments, by using transgenic experiments, we found that the Wnt secretion from the stem cells don't activate the proliferation of the stem cells, but the Wnt secretion from the PDGFR positive can activate the stem cell. So we have to understand that Wnt are a group of proteins. There are 19 Wnt, and many Wnt can activate this, but the Wnt which are expressed at this time may not be activating this. So we have to do further analysis in future to understand what Wnt are important in this proliferation to control the proliferation. So I'm not going into the details of more experiments, what we did to understand the more niches, but I will describe the result, what we got, and that is very interesting. And you can see the differentiating the niche, the dynamics of the niche changes, because in the proliferation we saw the PDGFR positive cells secrete Wnt, but the stem cell does not. But when we did some more experiments, while the cells are differentiating, we found that the stem cell can act as a niche for the progenitor cells and the differentiating cells, and can secrete Wnt. This Wnt from the stem cells can activate the differentiation of these cells into ciliated cells and club cells and other differentiated cells. But at that time, the PDGFR positive niche is silent. That means there is a dynamics in the niche, dynamics of the niche, how the niche changes during proliferation and differentiation. This is very unique, and if we understand this, we can control the proliferation versus differentiation defects in many cases. For example, in aging airways, we found that the niche is still the Wnt producing cells are the stem cells, and they invaginate into the epithelium somewhat like malignancies. And maybe this can be the reason why with age we see more tumors, more cancer. People are susceptible to more cancer with age. So this phenomena, which we published recently, is very interesting, and this has to be worked out in details how aging and lung cancer is related, whether this is related because of this. And so this paper came, we published this paper, and like the title was distinct spatiotemporal dynamic wind secreting niches regulate two proximal airway regeneration and aging. And I was a corresponding author of this paper, and this is the paper in cell stem cell. Then we wanted to know if beta catenin, wind beta catenin, is important for proliferation of the stem cell. What is happening with the lung cancer? And the lung cancer, as you can understand, is the most deadly cancer, and the estimated deaths is far more than as compared to other cancers in both males and females. So we need to pay attention to the lung cancer, especially with pollution going up and up. This lung cancer is becoming a menace. And out of that, the squamous cell lung cancer is the second most common subtype of the lung cancer, and after adenocarcinoma. So we know that the squamous cancer are initiated by the K5 positive stem cell, which I was showing earlier, and these are the progression of the tumor progression. How it progresses is from normal epithelium, and this is the hyperplasia, something like which I was showing in the earlier slides. Then this hyperplasia then leads to squamous metaplasia, then dysplasia, then carcinoma in situ and invasive carcinoma, where it leaves the basement membrane and go inside and metastasizes. So these are different steps, but in lung cancer, we don't know exactly what signal can activate this, what mutation can activate this, and the sequential pattern is not known. So we are investigating these also, but initially we want to know the evidence whether Wnt dysregulation can be important in the stepwise progression of squamous cell lung cancer. So this phosphoform which we discovered, Y489, is not reported much in the whole world. So we found it, and we found that it's not available in these are human patient samples, and this is a normal human airway epithelium, and you can appreciate that there is not much of the beta catenin available in the stem cell, but with human pre-malignant sample airway epithelium where the cells are dividing, and the green marks the stem cells and the white marks the progenitor cells. The progenitor cells and the stem cell both are dividing, so we can see that there's a significant amount of nuclear beta catenin because the beta catenin is coming to the nucleus and making the Wnt responsive genes active. So that means Wnt has a role to play in pre-malignancy. And when we tried to do with the lung cancer, squamous cell lung cancer, we found again very much increased amount of Y4 beta catenin, Y489 localization in the nucleus. So these values are quantified here, and we can see there is a significant increase in the amount of beta catenin accumulated in the nucleus, as you can see from this image and this image. And as beta catenin in more and more localized to the nucleus, it activates the Wnt pathway, which is important for stem cell proliferation, and therefore it can cause more and more tumor. So we also quantified the amount of beta catenin in the stem cells also, and we found the beta catenin in the stem cells also increasing. If we mark the K5 cell here, here, and here, then we see a significant increase, suggesting that this Wnt beta catenin, especially the beta catenin Y phosphoform Y489, is very important, and we should target it. So to better understand whether other Wnt secretion pathway is also important, so we again studied porcupine, which again activates the Wnt secretion. We saw that porcupine, like beta catenin, is active in the pre-malignant lesions and highly active in the tumors. And this is quantified here. You can see the increase in porcupine, suggesting that the cells in the epithelial cells as well as the mesenchymal cells in the around the tumor stroma is secreting porcupine, which is then releasing the Wnt. The Wnt then activates the stem cell proliferation or the tumor cell proliferation. So again, this is very significant data which suggests that Wnt beta catenin is active and probably driving the stem cell proliferation. So now the question is, how can we model this in vitro experiments? Because we want to do a drug screening, and how to model that. So we isolate stem cells from the trachea of a mouse, and then we culture them in submerged culture like this, and this is the day course, and we change the media. And here we use a chemical compound called CHIR. It's a inhibitor of the GSK3 beta, GSK3 beta. I can show you what was GSK3 beta. GSK3 beta was initially inhibiting the beta catenin and degrading the beta catenin. So if we stop this, this cannot inhibit the beta catenin, and therefore the beta catenin will be more active. So this is a way to activate the Wnt pathway, and we can see in this case the K5 marks the stem cells, and the K5 marks the stem cells, and the EDU marks the stem cell proliferating stem cells, the dividing cells, and we can see that with using CHR, the stem cell proliferation increases because the Wnt pathway is activated, and we can see a significant increase in the number of stem cells that is dividing. And we also checked with the CHR, can we get an increase of the beta catenin phosphoform, which we discovered Y489. And we saw that yes, CHR with DMSO, that is the control, we have normal level of CHR or beta catenin, Y489, but with CHIR, there is significant increase in the number of cells as well as significant accumulation of beta catenin in the nucleus, and the data is shown here. So this is the activation. We check the activation of this through the TCF-LEF reporter assay. This reporter assay says can give us an idea of how much Wnt signaling is active. So when we add CHR as compared to the control, the 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 activity is significant. That suggests that this CHR is a good compound to use and to model this as an in vitro model. We also used to check whether these can be used with other inhibitors, whether similar things can be happening. And if the CHR acts similarly with other inhibitor like GSK3 4, and you can see the data is here, which suggests the K5 cell number, which is the stem cell number, increases once we add CHIR or GSK3 15. So both these inhibitors can be used for the screening, but we got significant good data with CHR. And as you can see, this is the normal epithelium, but once you add CHR, they form like tumors on the plate. So we also cross-check this with this model with direct Wnt, which we added from outside, this Wnt protein. And this suggests that the Wnt protein can directly activate cellular proliferation and cellular proliferation. So this is a good model system to go for a drug screening. So what we did is we went for a high throughput drug screening, and we screened 20,000 compounds by using the TCF-LEF reporter assay, and in human cells, human lung cells. And we then did a secondary screen and then the tertiary screen to find out compounds. So we found a compound by this assay system using robotic screening system, a compound called WIC1. We call it Wnt inhibitor compound 1. And we got a multiple compounds, and these are now getting evaluated. And as you can see, this compound, when we challenge it with the CHR, as I said, is activates the Wnt signaling, and we wanted to check whether WIC1, which is the inhibitor of this pathway, can inhibit the function of CHR. So as you can see, CHR activates the stem cell proliferation, and then WIC1 reduces the Wnt pathway down. CHR activates it, and WIC1 inhibits it. So we found a molecule which can be used to reduce Wnt activity and therefore probably stop stem cell proliferation and tumor formation. So we checked whether the WIC1 inhibits airway basal stem cell hyperproliferation, and whether it can push the cells into differentiation mode. Because once differentiated, the cells will not divide. Usually differentiated cells don't divide. So it will not divide, and the cells which were undergoing hyperproliferation and pre-malignant like phenomena, they will become differentiated. So we use this assay on the human airway basal stem cells. So we isolated airway basal stem cells from the humans, and then added 2 micromolar of CHIR in one, and another was we added WIC1, which is our inhibitor compound. And you can see, as compared to it brought down the cell division to to the normal rate. So that means even if certain tumors have high amount of CHI, like Wnt activation, this compound can reduce the Wnt activation and restore normalcy to the epithelium. So now, can it differentiate also, help differentiate the cells? And we found that yes, as compared to it induces huge amount of differentiation, especially of the ciliated cells. And the ciliated cells are very important for maintaining the mucociliary protection of the airway epithelium, and you can see that it increases the ciliated cell formation. So this is a very interesting compound which can be used to restore homeostasis of the airway epithelium in pre-malignancy, and can be tried with tumors, lung tumors. So again, we wanted to know whether week 1 decreases nuclear beta-catenin Y489 and restores airway homeostasis. So we again used our compound 5 micromolar CHR to activate the stem cells, and then we added week 1, and this is the K5 cells and phosphobeta-catenin. The phosphobeta-catenin is shown here, and you can see with CHR, as we know, CHR activates the Wnt pathway, and Wnt pathway activates the beta-catenin translocation to the nucleus. So these cells are all beta-catenin positive for the nuclear staining. But with the weak 1, that is lost. That means it affects the important protein beta-catenin Y489 phosphorylation, this compound, and thereby is very interesting. And you can see the data here. The beta-catenin levels in the cells have gone down with week 1 treatment of the CHR. So it brings the Wnt level to the normalcy, and probably it will prevent from tumor formation. So this work was published in Cell Reports recently in 2020. And then I want to tell you all about how we are using the stem cell biology into COVID research. As you all know, the COVID causes is a severe pandemic and causes and the COVID virus affects the nasopharynx of a person and then the windpipe, and from there it goes to the lungs and affects the lungs. And the question is how to model this and study this is very unique. People want to know how we can model it to study it well. So there is no model system available to study this, but our lab is working on this type of models. We have been working on this type of models, and these are becoming very handy in doing the COVID research what we are doing. So as the trachea has cartilaginous rings, as I said earlier, so these epithelium can be cultured in a system called airway liquid interface culture system, in which we grow these human airway cells, and they differentiate under this culture condition. And you can see it here. The K5 cell is the stem cell, and the acetyl beta tubulin is the ciliated cell, which are the differentiated cells. And for the lower bronchial area, so these are bronchioles, and we can take these cells from here, and we can make lung organoids, which looks like this, using the inverse opal bead assay based scaffold co-culture system. And this system we published in 2017 and 18. We were the first group who engineered lung tissue, and you can see we used alginate beads and then coated with collagen. And after coating with collagen, we added iPSC derived fibroblast, iPSC or normal fetal fibroblast, human umbilical vein endothelial cells, small airway epithelial cells. We integrated all these cells together to form a lung-like structure. So as you can see, this is our model which we made, and this is human lung. So they look very similar in the number of cells and the architecture of the alveoli. They maintain it. So this is, we publish it, and it was covered in the news. So we can use this system very effectively to model COVID. So what we are doing is now using, so sorry, this is the adult human lung, and this is the engineered lung we made. So as you can see, this is pretty similar. So this can be a unique model to study COVID-19. And the other assay was air liquid interface cultures. So what we are doing right now using these two unique models, we are measuring the viral infectivity, viral replication within these cells. We put the virus on top of this, and then we see how many cells are getting infected, and we can do it in the differentiated as well as only stem cell populations. So this is the unique nature of this air liquid interface culture, and it can be easily done in many parts of the world. And if anybody needs the protocol, we can share it. And then we are also studying, after the virus infects the cells, what is the cytokine release parameters, and what are the other signaling, the immune aspects of these cells, how they interact with um, with the virus. And then we are using or planning to use this for drug screening. We are building a model for drug screening for COVID testing. And also we are studying the role of cigarette smoke on this epithelium, how the cigarette smoke affects this. So these models can be very useful. And we have I'm sorry, I cannot show you data here because they are unpublished data. And right now we cannot present the data here. I'm really sorry, but these are models can be used by many people in the world, and this can be very effective for COVID. So I would like to thank my postdoc boss, Professor Brigitte Gompard, Professor Volker Hartenstein, and Professor Stephen Dubinet for being patient with me. And this is our group who have published these papers. And believe me, this is the elevator, and we are all standing inside one elevator, and we took the picture. And I would like to thank Professor N. Sattamurthy, who was my director when I was a lecturer at ISR Mohali, and Professor Chinmoy Shankar De, who was my PhD mentor. And I was a little bit afraid because we were hit by magnitude earthquake 4.2 right away just before starting this presentation, and there were a lot of aftershocks. So I was ready to run if there is another earthquake coming. And so I will take questions now.
Thank you. Thank you, Dr. Pal, for such a nice and informative presentation. Uh, okay, there is some question.
Thank you very much for having me.
Thank you. There's some question for you.
Yes, please.
I have picked up from the chat box. First one from Aishwarya Rai wanted to know that if we take a active stem cell from pregnant woman's womb, then can scientists make any organ of that baby if it is needed someday?
So sorry. So what can you reframe the question? What stem cells from the is it you are calling about embryonic stem cell or placental cord blood cell, or what type of stem cell?
Oh, it may be placental stem cell, probably.
Yeah, I think with the technology with which we are having right now, we can probably program them into any type of organs, especially the embryonic stem cells. I have worked with those; they are very easy to work with, and you can modify them into different ways. And also, the placental stem cells should behave in the same way as like the embryonic stem cell. And now the techniques are available by which we can program them up to certain extent, which looks like organoids. Maybe total organ is not possible now, but it looks like organoids, and that can be done.
Okay, thank you. So another question from
Yes.
Arpita Banerjee. Can COVID-19 cause lung cancer?
I think the COVID-19 uh, causes a lot of inflammation inside the lung, and we know that inflammation is associated with lung cancer. So right now there is no data that COVID-19 can cause lung cancer, but with the amount of inflammation that happens inside the lung, there is a slight possibility that lung cancer may start, or at least if there are pre-malignancies already present, those pre-malignancies might flare up into tumors.
Okay, sir. Thank you. Thank you, sir. Sir, another more question from Shaheen Ahmed. Can stem cell research cure cancer?
Yeah, stem cell research has been used for a long time to cure cancer. Like bone marrow stem cell transplantation is done to treat blood cancer, right? So stem cell research has been used for a long time. Now other stem cell like types are being used now thought of to use. Like mesenchymal stem cells can be used to deliver proteins which can inhibit tumor formation.
Okay, okay, sir. We can take one more question. Sir, for you, one more question from Devaroti Mukherjee. Sir, if you can elaborate the role of CHR in restoring normalcy of epithelial cell, would be
CHI
helpful.
is a compound which inhibits GSK3 beta, and GSK3 beta, by inhibiting GSK3 beta, it activates beta catenin. So beta catenin once activated, it goes to the nucleus and activates the Wnt responsive genes. So CHI cannot restore this pre-malignant phenotype, but our compound, the weak one, can do that. Anything which can inhibit the Wnt signaling can do it.
Okay, thank you, sir. Probably Devaroti Mukherjee got his answer. Sir, another more question from Aparajita Banerjee. Is lung cancer preventable?
Sorry?
Is lung cancer preventable? We can
Yes,
prevent the lung cancer.
we
Can we
can
prevent
prevent lung
lung
cancer
cancer?
for sure. The main thing is the immune system of our body. If the immune system is strong, we can prevent it. And there are multiple immunotherapy regimens which are being used now to cure and prevent lung cancer.
Okay, thank you, sir. Thank you so
Thank you
much.
very much.
Now we have reached at the end of the first half of the second session. We are happy to have you among us today. Thank you, sir. Thank you, Dr. Pal,
Thank
and
you very
looking
much for
forward to
having me.
have you. Yeah, looking forward to have you among us in future. Thank you, Principal Madam. Thank you, IQAC Coordinator Madam. Now I like to invite Dr. Devdeep Mukhopadhyay to take charge for the next half of this session. Now it is over to Dr. Devdeep Mukhopadhyay.
Thank you, sir. Good evening and welcome everybody to our last lap of the webinar. Now let me introduce about our last speaker, Dr. Nandan Kumar Mondal. He is now working as an assistant professor in Baylor College of Medicine, Texas, USA. But originally, he is son of the soil of the University of Burdwan. He is undergraduate from Siuri Vidyasagar College and masters from the University of Burdwan in 2005 with gold medal, and also MPhil in 2007 from the same university. He has earned his PhD from Chittaranjan National Cancer Institute, that is CNCI, Kolkata, in the year of 2011. Subsequently, he was actively engaged in teaching in the Department of Zoology, Kobi Nazrul College, Birbhum. After that, he moves to USA for postdoctoral study and now working on heart failure, its transplantation and circulatory support. Today, he is going to deliver a lecture on the topic entitled Heart Failure, Biomedical Devices and Paradigm Shift in Heart Transplantation. So now I request Dr. Mondal to start the lecture, and we all enrich ourselves about our most lovable and adorable organ of our body, that is heart. So it's now over to Dr. Mondal. Can you hear me, Dr. Mondal?
Uh, can you see me? Can you hear me?
Yes, yes.
Okay.
Please
Thank,
start. Hmm.
Thank you so much for the nice introduction, Dr. Mukhopadhyay, and it's an honor to be a part of this international webinar. And oh, you cannot see my slides, right?
Yeah, not now.
So
Hmm.
okay, so I'll share it later. So today, I am going to deliver a lecture on the basics of heart transplantations, heart failure, and what are the biomedical devices. But before that, I would like to thank all of you, the organizing committee, the principals of the Hooghly Women's College, and mostly importantly, Dr. Mojumdar, who is effortlessly working with me since last couple of days to figure out some time frames, because we are all going through the COVID situations, and everything is and Texas is a hot spot here nowadays. So we are very busy nowadays. So ultimately, we got some time, and we are ready to go. So how to share my slides? Can you see my slides?
No, sir, till not now seen.
Okay. Okay, just a minute. A window, Women's College. See, you can see that?
Yes, it's coming.
Okay.
It's coming, sir.
Okay, so here you go.
Yes,
So
so please start.
today's topic is heart failure, biomedical device, and the paradigm shifts in heart transplantations. So before I go into the detail of my current topic, I'd like to mention the very basic parts of our human body. That is, what is heart and how it works. Everybody knows that, and what are the heart diseases, and what is heart failure, and how we treat the heart failure. So before that, I'd like to shortly a quick overview of the anatomy of the circulatory systems of the human body that I know that everybody knows, because they are either undergraduate or postgraduate students and leaders in the field. So I'm not going to elaborate these sections, but I would like to mention a little bit about the human heart is a muscular organ that provides a continuous blood circulations through the cardiac cycles, and is one of the most vital organ in the human body. The system is consists of a network of blood vessels such as arteries, veins, and capillaries. These blood vessels carry blood to and from all the areas of the body. Now, what heart is covered by a pericardium, a sac, the double-walled protective sac. It's called the pericardium, and the double membrane of pericardium consists of the pericardial fluid, which nourishes the heart and prevent shock. So this is the human heart circulations that everybody knows, how the blood comes from the superior vena cava, goes to the lungs, and then oxygenated and delivered to the all over the body, rest of the body through the circulations. And here the heart is the central pumping organ that can play the pivotal role in this process. So I have a quick video, but I think this is not relevant for this, but it's important. The video is not playing. Can you see the video? Why it's not playing? Okay, whatever. So now I'm going to the what are the types of the heart disease. So congestive heart failure, cardiomyopathy, valvular heart disease are among the heart disease types. There are many more than the 50 types of each of affecting different areas of the heart and the blood vessels, which but among the congestive heart failure develops over time. So what is heart failure? So in this picture, you can see a normal heart and the failing heart. So what is the difference between, you can see the weakened heart muscles that cannot pump enough blood. So that's the reason the heart is not pumping enough blood and body didn't get enough oxygen. So it's a dysfunctional heart. So heart failure classifications, so here are different way you can classify. I took this from the Indian journal, Heart Journal. So you can see there are many ways on the basis of the left ventricular ejection fraction, on the basis of the time course, and on the basis of the symptoms. So this is the classifications, the class 1, 2, 3, and 4, actually New York classifications of the heart failure. And here we follow most of the cases, this type of classifications in United States. So congestive heart. Oh, can you see the video? Because my
Yes,
presentation,
yes, sir.
but it's not playing.
Not playing.
Oh, it should be, because my presentation mostly on video presentations, but if you can't see it, then it's just a minute. So basically in this video you can see what is the congestive heart failure and how it occurs through the body, and there are many different conditions that can lead to the congestive heart failure in the heart attack, and the blood vessel, blood pressures, and the coronary artery disease. Any of these can prevent the heart efficiently pumping blood to the rest of the body. As a result, you can see the kidney sense. I can see the video. Can you see the video? No. Oh.
Still
Okay.
not playing, sir.
Okay, so maybe it says the media player is not there. So I'm moving to the next slides. So basically this is tells about the cardiovascular disease scenario in the United States. So cardiovascular disease is the leading cause of mortality globally, like the heart failure affecting 5.8 million patients in the United States. Fatality rate is 1 in 5, and fewer than 60% survives within 5 years. And the cost of the cardiovascular disease is around 444 billion. That is huge. Now, I was trying to figure out the cardiovascular disease in India, but unfortunately, you can see a lot of the empty spaces. The data is not available, so I cannot come into a conclusions what are the current scenario of the heart failure in India. But the heart failure is associated with shorter life expectancy, increased frequency of the hospitalizations, and poor quality of life, and is the major public health challenges even in India. So treatments for heart failure, we'd like to focus on the medications. So initially is the medications. This is not the current topic of my presentation, but there are so many medications that can initially be used to treat the heart failure or to maintain it. And the other treatment is the surgery or medical device. So this is the main part. So when a patient have a heart failure, and there are several ways to correct them or to completely change the heart. So to correct the coronary bypass surgery, heart valve repair or replacement, implantable cardiovascular defibrillator, cardiac resynchronization therapy or biventricular cardiac pacemaker, and ultimately the heart pumps, which is called the ventricular assist device. So heart pump is a small machine, as a metal made machines that can facilitate the circulations, that can facilitate in our mask, that can facilitate efficiently pumping the blood through the heart. And ultimately the heart transplantation, when is heart completely damaged and it cannot, so transplantation is the other options. So ventricular assist device, what is ventricular assist device? A ventricular assist device is a mechanical pump that used to support heart function and blood flow in people who have weakened heart. The device takes blood from the lower chamber of the heart and pumps it to the body, the vital organ. So when we can use the VAD during the surgery until heart recovers, or while waiting for the heart transplant, or in some older patient, it can be used for destination therapy. Suppose the patient is 80 years old and we cannot transplant, so we can put this LVAD device and leave over there for destination therapy. So can you see the video? I should check it before, because most of the presentation is including lots of fascinating videos. So here, what I am describing, you can see this is a type of ventricular assist device, and this is the older generation versatile devices, and it can connect to the heart and blood goes to it. If we have this video, we can see the animation how the blood comes from ventricles and plus to the aorta and circulates to the body. So it can facilitate. So this is the older generation and bigger size, and now we have so many modifications of the newer generations with continuous flow devices that I am going to show you later. So now have a basic idea type of ventricular assist device. So there are several types. So two types of heads are left ventricular assist device, which can be connected to the left ventricle, and right ventricular assist device. If we use the both at the same time in the left and right ventricle, we call the biventricular assist device that you can see from the picture. So this is the left ventricular assist device connected to the left ventricle, and this is the right ventricular assist device connected to the right ventricle. And here you can see the both devices are connected to the heart to facilitate the pumping of the blood throughout the body, to actually support the heart to pump efficiently through the body. So biventricular assist device. So VAD has two basic design: a transcutaneous VAD. A transcutaneous valve has its pump and power supply located outside the body. Tube connects the pump to the heart, to the small hole in the abdomen. The type of the VAD might be used for shorter durations during the heart surgery. So it's an outside, it's a big machine, so patient cannot move. And another is the implantable VAD. Implantable VAD actually inserted into the chest cavity, as you can see in the figure, and its power source is located outside the body. A cable connects to the pump to the power source through a small hole in the abdomen, and this is the normally currently we are using this. The older device we are not using nowadays. So if we look at the history of the blood contacting biomedical device, so as you can see, all these devices from generation first pulsatile pump, older technology, to newer technology, the third generation pump, you can see all are made up with metals, and blood is circulating through these. There is a pump. Every ventricular assist device has a pump, and blood goes through this. So blood has contacted with the artificial biomaterials. So, and there can be a damage to the blood, but that is not the, I will discuss this later, how we can mitigate this problem. But this is the life-saving technology. The first generation, the HeartMate, that is so big. Now we have the HeartMate 3 that recently we are currently using this HVAD and HeartMate 3 in our institutions nowadays for most of the patients. This is the newer technology. So I would like to say about our institutions because it caused the history of innovation. The Texas Heart Institute, if you see, we are the institutions who it was first established in 1962, and the first successful heart transplant in 1968 United States. First artificial heart implantation in 1969. Then the cardiovascular surgery laboratory established in 1972. After that, a second total artificial heart implanted for the first time. We are the first 1981, and the hemopump, first rotary blood pump implantations in 1988. The Bart Fraser, the prominent scientist, and the physicians. This is the living legend. He is still here. And 1990, we have the Jarvik, the first preclinical implantation at our institutions. Then the Dental A Cooley building established in 2002, and currently I am my office is in this building on the 9th floor. And the HeartMate 2, the implantations in the United States, first in our institution 2003, and now in 2012 we are trying to develop another new VAD, and this is a total artificial heart. Oh, can you see the video? Okay, total artificial heart, and it is very sophisticated, and Dr. Bart Fraser and Dr. Kohn continuously, tirelessly working on this to make this usable. And now, okay, so in this video you can see that a total artificial heart and how it works. This is the rotary total artificial heart, and the powerful, and the blood flows, how you can see the blood from where the blood is coming from the ventricles, and this is the internal component of the part, and blood flows, circulates, and then move to the different part of the body. So this is efficient motor, and the durable, it's a magnetically elevated mechanical wearing, and this is very interesting. And I'm not going to detail about the mechanism, how it works or everything. I just want to show you that how this kind of artificial device can work, and the blood can goes, and where is the gap, where is the contact between blood and artificial surface, and what is the frequency, or what is the limitation of certain device. It's a pulsatile, it can generate pulse, how the pump output look like. So I'm not going to, this is a long video, I'm not going to elaborate it. So in this slide we can say that the division of the cardiothoracic transplantation and circulatory support has been a leader in the world of transplantation and circulatory surgery. So here you can see Michael E. DeBakey. So development of the total artificial heart was initiated by Michael E. DeBakey in the Baylor lab in 1950s. So Dr. DeBakey obtained his first federal grant in 1960 to develop an artificial heart, and Baylor has remained the world's leader in the surgical treatment of heart failure. The first successful heart cardiac transplantation in the United States was performed by Dr. Cooley in 1968. And here you can see that Dr. Noone, Dr. Bart Fraser, and Dr. William Cohn, all three are currently serving as a senior faculty in our institutions. And under the leadership of Dr. Bart Fraser, a total of more than 1,300 heart transplant and over 1,200 ventricular assist device implant have been performed at our institutions. And this is the world record. Nobody did this in the world. So he is the pioneer and the living legend, still working at our institution, 80 years old. So why you said that we are the first successful pneumatic LVAD implant in US, 1963 by Denton Ecoli. And this is Dr. Bart Fraser at that time. And this kind of had, after that, there are so many improvements the VAD designs already have that I already mentioned to you. So we are first 1970, the bridge to transplantation LVAD. We are first in 1992. A patient discharged home on an LVAD support. 2000, the first in US to implant a Jarvik by ventricular assist device. 2003, first in US to implant HeartMate 2. So HeartMate 2, Jarvik, HeartMate 1, these are the different types of the ventricular assist device that developed over time with higher modifications. In 2012, the first US implant of the continuous total artificial heart and the Syncardia heart, total artificial heart, have been implanted. So we are the first, we are the leaders. I'm telling you because I'm very fortunate to be a part of these institutions where actually the artificial device designed and made. And since 1968, we are continuously making a lot of new technologies and doing a lot of surgeries. So survival with LVADs, if you look the Kaplan-Meier curve in here, so here I can say that HM2 means HeartMate 2. This is one kind of VAD, continuous flow device. BTT means bridge to transplantations. And here DT means destination therapy. So bridge to transplantation means when a patient have a heart failure, we immediately need a heart transplantations. But heart transplantation, we cannot do heart transplantation. Okay, you need a heart, we are giving you a heart tomorrow. No, you have to wait. You have to be in the waiting list. So once the heart is available, you will get the transplant. But since then, from that time point, from this time point to until you got a new heart, so that is called the bridge to transplantation. During that time, we'll keep the patient alive by the support of the ventricular assist devices. So you can see the survival is pretty good. So different types of and the LVAD durations, how it's and the Dr. Fraser's and what are the survivals of the patients. You can see here. So now I would like to say the current generation of LVADs used in most frequently in our laboratory. So the first is HeartMate 2. The product name is HeartMate 2, and it's a thoracic corporation device and approved in January 21, 2010. And the second device is hardware, is the newest one, is the September 27, 2017 approved by, and you can see the design of this device, how they work, and what are the specific gaps, and how the blood flows, what the motor looks like. So the first device is so there's different. All three are continuous flow. Some are axial flow and are continuous. All are the continuous flow devices, not the pulsatile. So they cannot generate the pulse, but they can continuously pump the blood. And the most importantly, this is the HeartMate 3, the newest device approved in October 18, 2018. So we are currently using the hardware and the HeartMate 2 mostly in our institutions. And here you can see the Texas Heart Institute or Baylor experiencing with LVAD. So more LVAD than any other program in the United States. So we did a lot. So BSLMC means Baylor St. Louis Medical Center historical VAD volume. You can see since 1986 to 2006, I have the data. So how many VADs and the survival rate, if you see the 2012, 13, 14, 15, 16, and 17 is 19%, which is higher than the national average. So Intermax is the all over the US about the survival of the LVAD patients. So we are still more than that. So the few decades later, the famed surgeon, I already talked about the Bart Fraser, continues the quest of an artificial heart. So 1968, you can see, then since then he's keep continuing the developing the new artificial heart, developing the new valve heart, designing the latest technologies, and now in 2019 he's 80 years old, and I'm very much fortunate to work with him and collaborate with him. This is the great opportunity for me to learn something from you and apply my knowledge to help to mitigate the bad graded clinical complications and other things. As I already mentioned that these are the devices. When the blood goes through these devices, it can continuously come into contact with the artificial biomaterial. So this figure is this is a HeartMate 2. You can see I showed you the previous slide, the HeartMate 2, how it looks like. And when the blood goes through, it can come into contact with the motor, with the rotor, and there is a chance of hemolysis, the blood damage, the platelet dysfunctions, leukocyte activations, platelet activations, and they can ultimately lead to the productions of different lead to the development of platelet damage or platelet dysfunctions and ultimately thromboembolic complications. But we are continuously working to mitigate those problems by improving or designing the new devices to minimize this contact. So the importance of basic biomedical research in the field of cardiothoracic surgery. So people are telling me that I'm a PhD. I'm a PhD in hematology from Chittaranjan National Cancer Institute. So people are asking me, so you are now a faculty in the Department of Surgery. So how it match? So match is that I apply my basic science research knowledge, the hematology knowledge, and work with the clinicians and surgeons and engineers to tell them that, okay, so if you put this device in the body, so this is the case, this is the shear stress your device generate that can cause the platelet damage, and the platelet damage can cause can be responsible for the bleeding, can be responsible for the thrombosis, or can be responsible or other factors, leukocyte activation. What are the basic molecular biological changes in the blood that can tell or educate the engineers and clinicians. So this is the reason. So how we can mitigate, how we can design. So we have to work together. The clinical surgeons, engineers, and basic biomedical researchers all work together for the betterment of patient management. Now I'm going to move into my second part, that is the heart transplant. As I told you that, when a patient has a heart failure, we need to transplant their heart, but we have to wait it. So during that time, we need to support the heart for the time being, bridge to transplantations or destination therapy in some case. So the LVAD is the biomedical device that can support the heart. That is not the transplant, but this device help. Now the heart transplant, moving to the next section. So everybody has a simple question. We need a heart. We need to find a donor. So who is going to donate? I got a heart attack. Now I need a donor heart to be transplanted into my body. Now then you need a transport systems. So suppose I'm in the Calcutta and I desperately need a heart, and one heart is available in the Mumbai. So how is possible? How is the transport system, and how long the heart can be viable? What is the time frame? So we need to take care of all these things. And now I'm going to little bit of I'm not going to detail about the surgical technique, but the heart transplant procedures, how it works. So this is the patient. The heart is removed first, and the donor heart is taken out and then attached to this. This is so simple. But if you, I don't know whether you can see that. So this is the video, actual video, how the heart transplant. First, we got the donor heart. It has to be wet. So we have a wet, and the highly skilled, dedicated surgeons and doctors in 8 to 10 hours of surgery. So first, this is the patient, actual patient. We take out the patient heart, and this is the donor heart. Now the donor heart need to be implanted in the patient. So first we need to stitch back all the arteries and veins, and then in proper place. And once we place the heart, we need to use the electric shock to ultimately actually start the heart to beat and functions. So this is a real video, and this is the case how the heart transplant is done. It looks like a simple. Let's take the donor heart and put it into a patient, and you're done. No, it's a highly skilled and Texas Heart Institutions, and they're founded by Dr. Denton Equally, the first successful heart transplant in the United States. He did. This is the guy. He did the first successful heart transplant in the United States in May 3, 1968 at our institution. And we consider this first on the basis of the months or years of survival measures. So I'm very fortunate to meet with him, not here, because I joined Baylor in 2018, and this the pioneer of the surgeon, Dr. Denton Cooley, he died on November 18, 2016 at the age of 96 in Houston. So now I was looking at the when the first heart transplant have been made in India. So I found this literature. So the first successful heart transplant in India was done in the All India Institute of Medical Science, AIMS, New Delhi, on August 3, 1994. And the patient survived for 14 years. And since then, we do have a lot of heart transplant. LVAD in India, we know the Fortis Hospital, Dr. Devi Shetty's, and all of that. But we do not have that much of data available to report. But nowadays, India is also doing the heart transplants and the LVAD. The only limitation of the ventricular assist device is its cost, because here in the United States, everything is taken care of by the insurance. But in India, like where the middle, this is very difficult to do that. So Texas Heart Institute and Baylor College of Medicine, so over 1,400 heart transplants. So these are all the patients with the heart transplanted, and the survival after heart transplant in US. This is the total US scenario. The ISHLT database, International Heart Lung Transplant, and the UNOS database. As you can see through the survival curve, totally 78,000, and the half-life is 10 years, and conditional half-life is 13 years. So no other therapy currently matches this long-term outcomes for the end-stage congestive heart failure. So this is the ultimate solution. So if you have a heart attack and if you have a heart failure, the ultimate solution is to transplant your heart. Get a donor heart and transplant it. It's easy? No, it's not that easy because of the availability of the donor heart. So if you see the international number of the heart transplanted, now the heart transplant year and location. So North America, Europe, and other, you can see the year 1990 to 2030. So it's pretty consistent. It's not increasing or not decreasing. It's a flat curve. And why is that? Because the heart transplant, we need a donor heart. So donor shortage, the all organs in the United States. The Washington Post says that an organ shortage kills 30 Americans every day. It's time to pay donor? No. So here you can see the patient waiting at year end. This is donor organ transplanted, and this is donor recovered. So how the gaps look like. So how do you increase the number of the donor? So currently 4,000 people in the United States are on the national transplant waiting list for a donor heart. Currently only used heart from individuals who have been deemed as a brain death. So there are two different types of death, the brain death and circulatory death. So we only can able to get a human heart as a donor heart if the patient died through the brain death, not the circulatory death. So procured to the DCD, so donation after circulatory heart has the potential to increase the donor rate. So if a patient died because of the circulatory death, that is the DCD, our US law do not allow us to take this heart because this heart is dead. That is the circulatory death. So we cannot take this heart. Now the focus is going on the DCD. Why not? Because the DCD hearts are almost thrown away. Nobody is using this. So if we can recondition, if we can make this dead heart alive again, then we can transplant it. So that is the idea of the DCD heart, and this is the way we can increase the number of the transplant heart transplant because and save tens of thousands of lives per year in the US, hundreds of thousands globally. So transport of donor heart to recipient for heart transplant. So how it happens? So suppose here if somebody have an accident, or if you are listed as a donor, organ donor, so if you have an accident, suppose you had an accident in the road, and then you go to the hospitals, and then you declared brain death, and then your heart is available to take. So there are organizations, different organizations, they take care of it. So they just took the heart and they just ship that heart to the hospitals, and then hospital, how they ship, they just take the heart out from the body, put it in the cooler with ice, and then it goes to the hospital when there there is an ultimate need, or somebody, a patient is waiting for the transplant. So this is a long process. This is step 1, 2, 3, 4, 5, 6, 7, 8. So from the site of the donor heart should be transported to the recipient hospitals, and then heart transplant should be done. And this is not easy. So we need to match this. So supply is a lot, because people are dying every day. People are dying. There are a lot of people. They are listed as a donor, but can you get all the hearts? No, because if the supply is done, only 4% donor produce the viable organs, because we need an intact live heart to be transplanted. Only 4%, and among the 4%, only 30% the heart are used. Those are the collected from the brain dead patients. It's brain dead patient we are collecting heart, not only in the US but India also the same thing, the brain dead patient, not the circulatory death. So that, and we also need to match this patient. So donor and recipient need to be matched. And the risk, so it's a time taking. So so from 1 hour, 2 hour, 3 hour, 4 hour, 5 hour, 6 hours, if we pass the 6 hours, the heart is unacceptable. So when you get the heart from a donor, we need to implant this heart within this time zone, mostly this time zone, to make this transplant successful. And we need to keep this heart on the ice, and this is the current thing. Once we get the heart, we put it in the ice and ship to the recipient hospital. Sorry. So this is interesting. You see, I think most of you have watched this movie called The Traffic. It's Jimmy Settles, and how it took 150 minutes to save a life. If you don't watch this movie, just watch it. Here you can see how you can get a donor heart, and then you have the Mumbai traffic, and then transport this heart by road within 150 minutes to save the life. So the concept is 150 minutes. The concept is not beyond that. The concept is that if you are late, then the heart can be damaged or die. So what is the donation after circulatory death? So donation after circulatory death, how it's happens. So the family would like to withdraw life support. So patient is in the hospitals, and he patient is in the intubated, and the family member would like to withdraw life support. Patient has a long recoverable illness or injury, has suffered in the neurological devastation, but has not been deemed brain death. So it is not a brain death. The family withdraw support, wait for circulatory death, and procure heart. So circulatory death means when there is no circulation, heart is not pumping, heart is not beating, heart is stopped. So this heart we can use. So how we can use? DCD heart transplant in USA are not being done for heart in USA and India due to the concerns. The concern is susceptibility of the heart to ischemia. So 5 hours versus 10 hours versus 24 hours, and liver, and 48 hours versus kidney. So 5 hours for the heart. So there are several publications stated that the donation after circulatory death in heart transplant may increase the donor pool by 20% in US. As you can see here, the 41.6% the brain death hearts utilized 2005 to 2017, and the logistic regression modeling identified additional atho DCD donors. So these DCD donors, these hearts, nobody using, these are thrown into the garbage. But still we can use this heart if we can recondition this heart. So this number of the patient who are waiting for the transplantation can get this heart. So US we cannot do that, but in United Kingdom, the legendary hospital, the Royal Praput Hospital, Cambridge, United Kingdom, they are doing this. And still now, maybe there are more than 50 DCD heart transplant. They do this for the DCD. So their laws and legislation, they allow them to include this DCD heart to be implanted in the patient. But how they do that? So I was very much excited. So okay, if we bring this technology from the United Kingdom and any other Australia, the other parts is that to our country, and then we can still save a lot of human. So that's why in the last year I went to the London, Cambridge to meet with the leaders in this field, Dr. Large, Dr. White, and Dr. Messer. They are regularly doing this DCD heart transplant in patient, and I went there to learn how to do that. What are the things, and I got several knowledge, and then I came back to the United States after the visits, and then so we developed the new project here. So what is this? This is called the heart in a box. So this machine is the Transmedics organ care systems. So this looks like a Xerox machine, but in this machine there are several parts. We can take out the dead heart and put it here to keep it alive. So you can see the heart is beating, the dead heart is beating inside the machine, and we can keep this into this machine and then transport whenever we want to take it through via flight or via by road, and this can prolong the duration of the support, keep the heart functionality, keep the make the alive of the heart components, the tissue, cardiac tissue. So OCS device makes it possible to reconsider heart from donation after circulated death donor that were previously considered to be unusable, mitigate damage, and recondition the heart. OCS is readily being used for lungs, but not for the heart. Now it's time to consider for heart in US. So for that, we got a $4 million grant in 2020 to conduct the DCD heart research at Baylor College of Medicine over the next 5 years. So we already started our experimentation. So this is the transmedic OCS, and we are doing the heart, how put the heart here, and how we can manage, how we can keep the heart alive for the longer duration of the time. So this kind of things we are doing mostly on the research purposes, because the time it allow us to keep the heart on the machine, the dead heart on the machine, keep alive. It's not that much long, but still it's 6 hours, 8 hours, and we are trying to improve through the knowledge of the basic science. So my objective is to keep the heart alive, not only keep the heart in the machine, but also provide the necessary nutrients, necessary all the molecules, the chemicals, the cocktails of the chemicals to nourish the heart throughout the support period. To make the heart functionality proper and the viable even after 24 hours. So our target is 24 hours. So the future of the heart transplantation, as we call the paradigm shift, we use the unused heart can be used through these machines. This machine is called the Transmedics, developed by the Transmedics. So the organ care system, OCS. We need the console of these machines. We need the heart, donor heart, and we can keep this heart in the machine. We need highly surgical skills things, so that we can get this heart from the donor and put it here. We need a dedicated anesthesiologist that can intubating the donor for the collecting the heart, and this surgical team we need to get this heart out from the donor and then put it in the machine. And here I can show you how it happens, how it occurs. So just see this quick video, then if you have any question, I will ask you. I will give you a chance to elaborate you. So here is the case. So what happened? So this is the case. Let finish the video, then I will tell you. So the heart can be transported to the helicopters, and once we get the heart, so this is the cooler. So this is the conventional technique currently we are using. So we need to get a donor heart and put it in the ice and take it to the laboratory for transplantations. And there is a gap between the time. So we cannot keep this heart for longer time. As we increase the time, the risk increases. So after 4 hours, 5 hours, 6 hours, we cannot use the heart because of the ice. Now with this device, that organ care system, heart in a box device, it's very easy. We can keep the heart. Heart is continuously beating inside, because we have we made it. We keep the circulatory system on. The blood is circulating. We have the blood inside. We have the heart here. So outside of the human body, heart is beating in a machine. So this is the real heart beating in a machine. So heart is alive. Heart is happy. It's not a dead heart. It's a dead heart, and we made it alive. So our objective is to include all the nutrients and all the necessary necessary materials to support this circulation. So this is the way we can do it. So the transmatics is the new innovation. So that's called the paradigm shift. We can keep a longer and longer and longer time to keep the heart alive, and we can easily take it to the patient who needed to be transplanted. So suppose as you know that suppose in the Hawaii, every year, every time the Hawaii, we have a lot of donor heart available, but we cannot take this heart a total waste, because from Hawaii the 6 hours range, 6 hours range we cannot, but with this device it's a 12-hour range. We can make it, we can get this heart to somebody else. But and if we increase it more than 12 hours, we can take the heart from any part to other part for transplant eligible. So the ability to safely transplant a donor heart from DCD donor could be a paradigm shift to potentially increase the pool of viable donor organ to help more patients suffering from end-stage heart failures. And we are continuously doing our best job how it can be done in the United States, and we got this project recently. So hopefully it can be done very soon, and we can save a lot of patients' life in near future. Thank you.
Thank you, Dr. Mandal, for the fabulous and mesmerizing lecture. And now it's time to the interaction, because more and more question is there. First question is from Ayon Das. Donor and recipient can be how matched in case of heart transplantation?
Okay, so there. Can you hear me?
Yes, please.
Okay, so there are several criteria. There are because on the basis of the HLA we need to test the HLA human leukocyte antigens and the blood groups and other parameters. So this is already predetermined. And when you get the heart from the suppose a life gift, they have a available heart, so they have their parameters set. What are the parameters? What are the echocardiography? We have our own exclusion and inclusion criteria. Each hospital so they also set up those, and we need to match this. We need to match the transplant, because the transplant there is a chance of rejection. So we need to human leukocyte antigen matching study to do, and then we have to determine that this heart can be suitable for these patients.
The next question, what is the duration of VAD, and can we replace the VAD in case of SA node?
What is VAB?
VAD.
What it stands for? VAB?
VAD.
Oh, VAD, VAD. Okay,
Yes.
so can you repeat the question, please?
What is the duration of VAD in blood, and how is it replaced in case of SA node or AV node like this?
So ventricular assist device, as I told you, it's a support device. So it when a patient have a heart failure and suppose the left ventricle, if the left ventricle cannot pump enough blood, or the right ventricle, which any one of them, either left part or the right part is not working properly, cannot pump the blood, we use for the left ventricle, we use the LVAD, left ventricular assist device. So we can attach it to the heart for, and how long, that depends, and also depends on the pump speed. We have to modulate the pump speed based on the patient criteria. That physicians they are continuously monitoring the patients, what should be the pump speed, when to increase the speed, when to decrease the speed, and how long. That means how long means suppose based on the condition. So if your ejection fraction less than 10 and you immediately need a VAD implantation, or we put a VAD, and then patient will be in the hospital for approx 14 to 28 days, and then got discharged to home, and they have a drive line outside the body to connect the battery source, and they have the follow-up visit to come and clean the so prevent any infections or anything. So we can keep this LVAD until your heart is fully recovered and start again proper functioning. Or if you enlarge the patient, we can keep this VAD for bridge to transplantation. Suppose you desperately need a heart transplanted, so during this time period until you got a heart, we can keep the VAD over there. and for some patient you are very old and we cannot transplant it, we will keep it until as long as you are alive, and we need to closely monitor, because we do have the outside modular, we can check it, we have to see how the speed, we have to modulate the speed, that each device is different, because as I told you, the different company have different device, the HeartMate 2, the HeartMate 3, the HVAC, each has their own setup, and we have to follow their guidelines to maintain it.
Can artificial heart maybe acting as real heart? And another question is what's the biochemical parameter in case of blood when transplanted
Okay,
with
this is a good
artificial heart?
question. Yeah, artificial heart. Artificial heart is suppose nothing can replace a real heart. Real means real. Artificial mean artificial. Made up with biomaterials. So once we have a biomaterial, once we have a machine, that a machine cannot take place the real part, but it can assist you. It can keep you alive with some limitations. So suppose you have an artificial or whatever the blood which goes through the machine, and there is a pump, and there is a gap between the rotor and the machine, so the blood can pass through this narrow gap, and based on the gap the machine is rotating, and it can generate the high level of non-physiological high shear stress, and that shear stress can be responsible for platelet damage, mostly the platelet, because the bleeding, because as we can see that when you have a LVAD, the most important or most devastating clinical complications is the bleeding, infection, and stroke. So related to the blood clot or thrombosis. So blood related. So biochemical parameters means you can measure the platelet functionality, because the platelet have the receptor, the von Willebrand factor receptor, platelet have the fibrinogen receptor, platelet have the collagen receptors. So all platelets have these spikes. So when the non-physiological high shears, when it exposed with this artificial device, they can set the receptor from the platelet surface. So platelet without this receptor. So if there is a clot, if there is something, so how bleeding occurs? If the platelet lost all these receptors, or some of the receptor like von Willebrand, they cannot interact with each other. So they cannot initiate the clot. So bleeding. And another thing, if the platelet receptor like the pre-selectin, if it activates, then what happens? Then platelet-platelet interactions is more. So platelet aggregation occurs. That can cause thrombosis. So thrombosis and bleeding, these are simultaneously can occur, because the non-physiological high shear stress can be responsible for bleeding, can be responsible for thrombosis, and it's a paradoxical effect. It's a paradox. Nobody knows that is only the pad is responsible for this kind of clinical complications, or the patient has the pre-existing conditions, because each patient is different. I am different. How I will respond to a pad, that depends on my body, on my ability. And if you compare with myself with another guy, each individual is different. And the parameters, mostly we are looking at the platelet functionality, leukocyte activations, and we also look at the serum plasma, because if the platelet got deactivated, platelet got activated, we all have the blood markers for these all receptor, GPIB alpha, GP6, GP2B3A, CD62P, pre-selectins, PAC1, integrin, all these kind of things we can measure. And also the damage associated molecular patterns, damp generation, high shear stress that can generate the damp, the ROS production, reactive oxygen species. ROS is the important factor. So we saw the ROS, higher level of ROS, and minimize the antioxidant and superoxide dense SOD, catalase. These are all the parameters, all the biochemical things that we can measure in this patient. If you see my publications, I do publish a lot, more than 10-20 papers on the platelets, the leukocytes, how it led to the bleeding, how we can mitigate this. You can search in the PubMed. So I haven't mentioned this because this topic is only 1 hour, so I want to restrict this as much as possible. Mm-hmm.
And the last question, Dr. Mondal, that is much more interesting. In case of heart transplantation, if diabetic patient is also favorable, or because the diabetic patient has the immunocompromised, and the another part is the COVID-19 affects any case of heart failure or myocardial infarction. Please explain.
The pre-existing conditions patient, or because heart failure is mostly is the elderly case. Yeah, we do have a young patient got the heart failure with no comorbidities or no other pre-existing conditions, but mostly the aged, who are the aged person, they got the heart failure and then they desperately need a heart. So pre-existing condition is always there. Yeah, we have diabetes, we have hypertension, we have cancer, we have prior cardiac surgery, we have multiple surgery, and we have pacemaker, we have lung disease, we have COPD. Yeah, all this because these are elderly patients. So in all these conditions, the physicians, they knows how to manage this patient. If you need a heart, if you need to be transplanted, if you need an LVAD support, and they knows how to manage these patients. They knows how to adjust their drug, how to adjust their, and if you're hypertensive, how it can affect. If you have the high cholesterol, if you are diabetic, how it affects the outcomes. These are all. But whatever the pre-existing condition you have, based on the criteria, the physicians, they, they implant the device and manage the patient accordingly. So this is the first. What was the next questions?
And then the next is the COVID-19. How affects the COVID-19 the virus in case of heart failure?
In case of this kind of because we are currently experiencing a lot of COVID-19 cases in our hospitals. So every day, so we have a new building also, Magnet Campus. They also open for COVID testing, and at our institutions, we are very restricted. Every day we come to the lab, we have a face shield, we have a mask, we have everything sanitized, and then come here because there are patients who have been admitted for transplantation, have been admitted for heart surgery. Some of the patients are positive for the COVID, tested positive. Some of the asymptomatic, some of the symptomatic, but still. And if you have the implantations, how you can manage? So this is the challenging thing we are facing. We are experiencing a lot of patients in case of this. And if you see the literatures and then how the ECMO can be supported, how the ECMO can help the COVID. So actually this is not the current focus of my research. So I am not currently dealing with the COVID patient, but yes, we do have a lot of patient. We do have a patient with this post transplant or the post implant patient who have COVID, or the patient who come to the hospital with COVID need this kind of surgery. And the physicians, they are managing this according to their protocols, whatever is available nowadays.
Thank you, Dr. Mondal, for the fabulous and mesmerizing lecture. So now it's time for the valedictory session and vote of thanks. Now we turn to the Ms. Shreya Bhattacharya for this session.
Thank you so much.
Thank you. Am I audible?
Yes, you are audible. You can continue, please.
Thank you, sir. Thank you, sir. We have an excellent evening today. Finally, we have come to the end of the one-day international webinar organized by Life Science Department's Hooghly Women's College with support of Internal Assurance Cell. First of all, I would like to thank our Honorable Principal Madam of our college, Dr. Shima Banerjee, for gracing the webinar and delivering the inaugural address. Thank you, Madam, for your inspiring speech. Without her tireless efforts, this webinar would not succeed. I would like to express my sincere gratitude to our honorable speakers, Dr. Shauradipto Ganguly and Dr. Promit Das, both from Department of Medicine, University of California, San Diego. Dr. Manoj Kumar Paul from Department of Pulmonary and Critical Care Medicine, University of California, Los Angeles, and Dr. Nandan Kumar Mondol from Department of Surgery, Baylor College of Medicine, Houston, Texas, for their excellent presentations and making this webinar interesting and meaningful. We are really grateful to all of you for accepting our invitation and spare times from your busy schedule. We would like to thank Dr. Shamolina Goswami, IQAC Coordinator of Hugliomal College, for her enormous help in organizing this webinar. Thank you so much, Madam. We are grateful to Professor Satyajit Biswas, bursar of our college, for his active support in organizing this webinar. Thank you, sir. Our most sincere gratitude goes to Professor Chirantan Hajra, joint convener of this webinar, for his efficient technical support in organizing everything so perfectly and pouring confidence in us to make things happen. Sir, we really appreciate your great effort that has made us successful in this venture. Last but not the least, an educational webinar can never be successful without diverse participants. I extend my heartfelt thanks to all the dignified professors, research scholars, and students of our college, and as well as other colleges and institutions for making it beautiful and successful. With this, on behalf of the organizing committee of the webinar, I, Shreya Bhattacharya, Department of Nutrition, along with all my beloved colleagues from the Department of Botany, Zoology, Microbiology, and Nutrition, thank everyone and declare this webinar as over. Thank you all. Thank you very much. Wish you all healthy and prosperous life. Thank you so much.